Good morning, sir, ma'am, miss. My name is Felicia Diputri, and today I'm going to share what I've learned this week. About 250,000 babies born with cleft lip with or without cleft palate. About 50% of cleft palate and 10% of cleft lip and palate are associated with syndrome such as Van der Waals syndrome, craniofacial microsomia, Down syndrome, and fetal alcohol syndrome. Stickler's and velocardial syndrome are the most common syndromics associated with cleft lip and palate. The factors associated with this condition such as genetic or environmental factors like smoking, maternal diabetes, obesity, and exposure to alcohol. Embryonic phase development begins at week 4 after conception from neural crest, ectomesenchyme with 5 prominent cysts, frontonasal, maxillary, and mandibular. Then, during 8th week, if there is a disruption in the fusion of 3 embryonic components, the delay in the elevation of palatal shelves from vertical to horizontal, defective fusion, or past fusion rupture can occur. Cleft lip and palate treatment not only focusing in birth defect, but also surgical, speech, hearing, dental, psychological, as well as cognitive. The abnormality of any structure or function from birth called birth defect. Orofacial cleft is recognized as the disruption of normal facial structure that occur when the embryological fusion, the lip, alveolus, and/or palate is disrupted. This condition can be described as syndromic as well as non-syndromic. Environmental factors in orofacial cleft are maternal environmental factors, socioeconomic status, maternal disease, or drugs. Orofacial cleft can be prevented with medical control, lifestyle, behaviors, medications, healthy body weight, and family history checking. Development of the face involves complex events during embryogenic development. The disturbance during this development can result in failure to meet and fuse to form the appropriate structure and deformities. Cleft lip and palate are the most common congenital anomalies of head and neck region. Lip is a pair of structure that presents anteriorly on the face. A mobile musculofibrous fold surround the mouth, covered externally by skin and internally by mucous membrane. Facial development occurs between 4th and 12th weeks, and by the 10th week, the face already has a human appearance. Genetic and epigenetic factors play a role in causation of cleft. Cleft was caused by gene expressions or environmental triggers such as mother's health, smoking, infection, pollution, or medication. The genes will control development by controlling cell division, cell differentiation, or morphogenesis. Genes can be affected by mutations, mutagens, and epigenetic factors. Face is derived on 4th week around the stomatodeum, meanwhile primary palatum derived on 6th week. Van der Waals syndrome is the most common syndromic form of cleft palate, which characterized by lower lip pits. Nutrigenomics related to the causation of cleft is folic acid pathway deficiency. The study of genes called genetics. Genes are hereditary elements containing biological information transmitted from parents to their children. The different versions of genes called alleles usually caused by the variations in DNA nucleotide base pairs. This variation in nucleotide base pairs also lead to variable phenotypes. Genetic code is the language in the DNA that carries the instruction required for making protein products. This genetic code divided by cells into triplets called codon. Codon ATG will dictate the cells to start reading, meanwhile codon UAG dictates the cells to stop reading. There is a condition called single nucleotide polymorphism. It was a germline substitution of a single nucleotide at a specific in the genome. This condition occurs every 100 to 300 base along the human genome sequence. Genome-wide association studies, or GWAS, was used to scanning the genomes, looking for genetic markers that can be used to predict the presence of disease, identify SNPs across the genome that are associated with disease or traits, and identify which genes that influence disease risk are involved in human disease. Exons are protein coding regions of gene. These exons will gather up making exome that make up about 1% of entire genome. About 85% of disease-causing mutation are found in exome. So that's it from me. Thank you so much for the attention.